Thank you all again for joining this session of Shadow the Scientist. My name is Jamaica Marshall, and I am again very pleased to be with you. And as the programs manager of the Shadow the Scientist initiative, it is a pleasure to bring to you again Mikala Capage and Jessica Bonnar to continue what they started with you yesterday. Ladies, please proceed.
All right. Thanks, Tamika. Okay, so when we left off from yesterday's lecture, we had gone through all of the different steps of an immunofluorescence assay. And we left off that final day on day 4 of the protocol, which is to do the imaging. And so that's why we are in this different room than we were in before. And Jess, I'm gonna have you turn the camera so that the students can see. This is the microscope that we're going to be working with today. And then if you can get a little closer, Jess, the microscope slide is located under here, if you can see it.
Can people see it?
Yep, looks like we can see it pretty good, yeah.
So
What's the estimated cost of this microscope? This looks like super complicated and super, technological, so.
yeah, that's actually a good question. I think it is probably around for this extra fancy box that costs a little bit more, but the base microscope is about 2 million.
2 mil, oh my God. Oh my God, my knees just went weak.
Yeah,
We got a question here. What's your question?
Why this microscope over like a standard?
So the question is, why this microscope over the standard one? And so the reason why for this microscope is because it has a laser in it that is shining on their sample and what they're doing, and that's how they're able to detect the immunofluorescence from it.
Yeah, so that's a great answer. Basically, like other microscopes that we would call light microscopes, just use like the same light that's coming from these like light bulbs on the ceiling to illuminate your sample. Basically just allow you to see it. But this is a special type of imaging called confocal microscopy. And it uses lasers to excite the fluorescent tags that are on those secondary antibodies and then collect the light that those tags are emitting. And that is just a really complicated piece of machinery in order to do that. And so hence the sort of fancy specifications of this scope. Yeah.
And I will also say there are fluorescence confocal microscopes that I've used as well that are like cheaper,
more normal, yeah,
in the hundreds of thousands as opposed to millions. But another thing that's I think very special about this microscope is the degree of magnification and the automation that it enables as well. That Mikala's going to be showing us today.
Yeah, the automation is also that's a really big part of it too.
Yeah. I think you'll see the complexity of the experiments that Mikala is doing are completely require automation and make it that part really necessary as well.
Right. So this is like a shared microscope too. So like, when you're done using it, other scientists can use it too. And
No, this is Mikala's microscope.
It's my
only.
personal.
Oh, wow.
I know,
we're just kidding.
but this is a microscope that only my lab uses. So sometimes in research institutes, microscopes will be shared between a couple of different labs, which is a great way to, like, the way it works is that a lot of labs will pay in to like funding the microscope and also help pay for its like maintenance. But my lab uses this scope probably like 12 hours a day, typically like honestly 7 days a week, because there are a lot of people in my lab and we all do this type of imaging. And so because of how much we use it, we are actually like able to have our own.
In fact, as we passed someone in the hallway, Mikala went, "So you for sure need to be on at 3 p.m. And they went, "Yep." And she went, "Okay."
This is a very nicely shared resource, but it is like it allows us to do all of our experiments, so we all like using it.
Nice. I think I also had another question here. Were you able to hear that question? No, okay. So she asked, like, do you guys only have like one of these $2 million microscopes, or are there like other microscopes throughout MIT that you use that are also like equally expensive or more expensive or around the same ballpark number?
Yeah, that's a really good question. So sometimes you want different microscopes to that are sort of specialized for different types of samples. And so, in addition to this microscope, my lab actually has a couple of other very expensive microscopes as well that we're able to use. And then there's this whole other type of imaging that I'm going to touch on really briefly. So the imaging that I'm doing is using light to illuminate samples, but we also do, not me personally, but people in my lab and many people around MIT do use a technique called electron microscopy, where you're actually using electrons to illuminate your sample. And this is really Jess's window of expertise. And those microscopes are insane.
Well, so, and I will say actually with Jamaica, we presented it at an AP chemistry class at Mount Whitney on Monday and Tuesday, where we talked them all through electron microscopy and the principles of electron microscopy. Mikala is looking at labeling cells with this microscope. With electron microscopy, I'm looking at defining the way that atoms connect to each other in space to build proteins. And so you're actually, rather than shooting beams of light with lasers, you're shooting beams of electrons in a vacuum at samples. And because electrons have a smaller wavelength, they allow you to have higher resolving power. Those microscopes range anywhere from $1 million to $6 million. And they are also used around the clock. And actually, that's shared between labs. It's such an important resource. Yeah. And I think my lab pays $200 an hour to have access to using those microscopes.
Wow. Wow.
Yeah.
I mean, this equipment is like crazy.
Yeah.
Right?
Yes. State of the art, like the most advanced thing that science
of
has.
Electrons and things, but I will say actually the Nobel Prize, sorry, and I will stop talking about electron microscopy after this, we're talking about light microscopy. But the Nobel Prize in chemistry was awarded in 2017 for electron microscopy for just like figuring out the fundamentals and how it works and getting it to a place to where we can image cells and proteins. One of the Nobel Prize winners, his life's mission for the rest of his life now is to make electron microscopy cheaper and to build microscopes that a lot more institutions can afford, so electron microscopy can be used more commonly around the world because it's a really, really important tool. So that is something that people are thinking about, and as we talked about yesterday with the antibodies, like a lot of expertise and time and resources went into these things, and so we do need to pay what they're worth.
Yeah.
Exactly.
Yeah.
Awesome. And then I think we got another question over here.
Is it like nerve-wracking or stressful to be working with like really expensive equipment like that? Like, are you worried about like, oh, what if I mess it up or if I break it by accident? Like, does that stress you out?
Would you mind repeating that? Sorry, we have a vacuum in here today.
Okay. I'm just saying, like, when you're working with like such expensive equipment, like, does it stress you out having to work with that? Like, if you accidentally like break it.
Yeah, all the time. I'm stressed all the time.
Both because of the equipment and not because of these.
Yeah, not because of what I'm doing right now, not because of you guys, but yeah, definitely I definitely do my best to treat this instrument with respect, and I went through people in my lab spent several hours training me to use this microscope, and I used it with these senior people in the lab for the first couple times, and then I was eventually told, okay, you can now go and do it yourself. But definitely whenever I'm using this machine, I am sure to be very focused and make sure that I'm hitting the right buttons in the right order and everything, because yeah, I would hate to damage it and cost my lab, money, but I'd also hate to damage it and make sure that the person who wants to do his experiment at 3 p.m. today, can. So I definitely, I'm very aware when I'm doing this. So
yeah, and that's a big thing in science in general. These things are so valuable, and respecting the fact that they're valuable means you have to be careful. You also inevitably make mistakes.
Yes, and you have to, you can't be afraid to use it, right? You can't be so afraid of breaking it that you just don't use it. So
yeah, and yeah. I definitely, I didn't tell you about this. I left a lab fridge accidentally open a crack over this weekend
Oh.
and damaged some reagents that people had built up. Not
Yeah.
a -80, fortunately.
Yeah,
It was
that's fine.
okay. Yeah. The -80 is really bad. And
Yeah. It happens.
and the easy thing would be to go, oh, I don't know who did that. Like,
Who can
really
say?
unfortunate you can say. And I was like, no, it was probably me. I was here late on Friday and I probably left it open, and let me help you remake those reagents. So
Yeah.
you will inevitably mess up, and like admitting that and claiming accountability and like helping people out in the best way that you can is also really important.
Yeah.
Hopefully you don't damage $1 million instrument. That would be really bad.
All right, so speaking of this really fancy equipment that I have sitting right next to me, I'm gonna get started using it so that you all can see, some of these images be produced in real time. So, as we just said, like I have to be kind of focused while I'm using the microscope, and so I'm gonna let Jess maybe answer some of the questions from the students while I do some stuff on the screen.
And Mikala's going to let us know when she's available again to answer more questions, especially about her science. But thank you for the questions that were so great right off the bat. Those are awesome. One thing that, also thank you everyone for filling out the feedback form yesterday. Also, we are now, Mikala's screen sharing with us, and we're now seeing the software that she's using to image her cells. She's going to describe to us what those cells look like in greater detail in a minute. But I do just want to point out that what she's imaging right now is the DNA of the cells, and I don't know if you remember from her slides yesterday, but we can't tell the cells apart right now. So she's looking at the larval ovary and all of the cells that exist within it, and all cells have DNA, so we can't tell specifically those egg stem cells apart from the non-egg stem cells in the organ right now. But she is switching to a new color, which is of traffic jam, which is that protein that is present in the nucleus of some non-stem cells. And she'll be able to explain to us in a little bit more detail in a minute what that is. Very cool. And here we have HDS. And to explicitly explain what's happening on the microscope right now, Mr. Woodbury, I think, mentioned it a little bit earlier. But specifically, right, so this is our fly ovary. Day 1, Mikala dissected the larva ovary. Day 2, she added the primary antibodies on. Day 3, she added the secondary antibodies on. And we talked a lot yesterday about the secondary antibodies and the fact that they have a fluorescent dye attached to them. That fluorescent dye is what is lighting the cells up these different colors. And specifically what's happening with the microscope is it is shooting a laser down at this sample. That laser is shining at a specific wavelength. The fluorescent dyes, I maybe got a little bit too excited when I was talking about the resonance structures of those, but when the fluorescent dyes are being hit with this laser, the fluorescent dyes basically convert it into a slightly different wavelength of light. And that different wavelength of light then gets passed on to I call it the detector because that's what it is for electron microscope, the detector underneath. And the detector is then showing us those images that we're seeing on the screen. So laser, fluorescent dye changes the wavelength of that laser. That wavelength then gets relayed and captured into the instrument software.
Yeah. Okay, so I've done the work to kind of set up the experiment for a particular larval ovary, which is what's on the screen here. And essentially what I have done is just find the sample, zoom in to make sure that the sample is sort of like correctly positioned in the frame of view. And then the type of file that I'm going to collect is something that we call a Z-stack. And what this basically refers to is that I'm going to be taking, or the microscope and the computer working together are going to be taking an image, one image of the larval ovary, and then moving down a very, very small distance, taking another image, moving down, taking another image. And so we call this a Z-stack because the images, we aren't moving in X or Y. We're staying in the same coordinates on X and Y and just moving from the top of the sample to the bottom. And each image is so basically it'll take an image, it'll move a little bit, it'll take another image, and that distance that's being moved is 0.25 of a micron. So a micron is 1 times 10 to the negative 6 meters, or 1/100th of a centimeter. One
1/1000th of a centimeter,
Yeah.
1/100th of a millimeter.
Yeah. So these are really small distances. And part of what Jess mentioned is why part of the reason this microscope is so expensive is because it has all of this like machinery that will very accurately move a quarter of a micron, which is like an imperceptible distance. Like
It's a quarter of a 1/4 of 1/100th of a millimeter. Yeah.
That's it, like that's way too small. And so you need all of this kind of machinery to like do that for you. So this microscope is going to, once I hit start, it's going to take 207 images, each one a quarter of a micron away from the other. And it's going to start doing that. So I'm going to hit start now. And what you're going to see appear on the screen is the very top of the ovary. And so it's imaging, and as you can kind of see it like flash on the screen, and that is an image being taken, the computer moving, an image being taken, the computer moving. And so it's doing this very rapidly. But because we have to take 207 images, and we have to do it for 4 different colors, it takes a little bit of time. So now that my acquisition is going, I'm actually going to be turning off the light. I know that we look a little spooky,
Crazy.
but it's better for the quality of the sample if you are imaging in the dark. So what we're seeing here is, I think Jess mentioned, just the DNA of the sample. And like I said in our lecture yesterday, you can't learn very much about the ovary just from the DNA. I don't know where the egg stem cells are. I don't know where the non-stem cells are. And so what's going to happen is it's going to take the entire ovaries worth of images just looking at the DNA. The microscope is going to make some noises. It's going to then switch, we call this switching channels, and it's going to actually then image the traffic jam signal in green, the HTS signal in red, and finally the VASA signal in pink. And so it's going to take 207 images 4 different times.
And actually switching channels is just switching to a different laser, as well as switching to a different wavelength at which you're collecting.
Yeah. So while this acquisition is running, if you can see at the very bottom here, it's keeping track of how much of the experiment has been completed. And this is going to take about 10 more minutes to acquire. And in that time, I don't really have anything to do. I just kind of sit here. So this would be a great time for any additional questions from you all.
I actually really wanted to highlight in the feedback that we got from people yesterday, one thing that people wanted us to talk a little bit further about is the context of this experiment. So not just that we're counting cell numbers, and I think we talked a little bit that we're interested in those cell numbers changing, but like really zooming out, what is the 10,000 foot view of this?
Yeah.
And then once Mikala goes through that, give us more questions.
Yeah.
But we want to cover that first, I think. That was a big commonality.
Yeah. Fantastic. So Jess, can I actually pull this a little
Yep.
closer?
Absolutely. Are we okay? I bumped the microscope.
One sec.
Oh no.
Yeah, it should be okay.
It's okay?
Yeah.
Okay.
Okay, so let's see. So what did we so when we left off from our lecture yesterday, I spent a little bit of time telling you guys that I was interested in understanding the genes that control the number of egg stem cells. And I told you about one gene in particular that regulates how cells are capable of contacting each other, and that I was going to be removing the expression of this gene from the system, and I wanted to see how that changes the number of egg stem cells. And the reason that I'm interested in understanding which genes control the number of egg stem cells is because we don't have a complete understanding of how the organ maintains maintains and knows how many egg stem cells to make. The way that this works is that the organ actually starts in development with just a handful of these cells, like 10. And by the end of development, it has like 150 of these cells. And that happens very, very, very consistently every single time. But we don't have a good understanding of why or how it knows to make 150 cells. And so I'm interested in how this process is controlled. So actually I need 1 minute to look at something on the scope.
Thinking about the big picture, like what questions do people have, like even further about like the 10,000 foot view of why we're doing this, or why Mikala's doing this?
Anyone had any general questions on that? Yeah, I think we kind of got a question over here.
Cool.
So I know that you're, like you mentioned, you like switch around or remove genes that you think might be responsible for the, I guess, change in the number of eggs. So like, what kind of like, I guess like, what's like the thought process you go through of like picking which gene exactly to, like edit or change? Like, why do you think that one's?
Great question.
Yeah, like how did I land on this gene that I'm interested in testing, and why am I not testing another gene, for example? Yeah, it's a really good question. I'd say two things. So one, part of the, I'd say one thing to start, which is that Drosophila melanogaster as a model organism is really, really, really powerful because you can test many genes at one time. And you can test many different genes in one experiment. And so the lab that I'm in, my boss is actually pretty famous for these just giant experiments where she will just test, or where people who work in her lab will test just like 150 different genes. And you can get away with this because Drosophila grow so quickly, they're so cheap, all of that. So part of the answer is that you don't necessarily need to have a really good reason for testing all of your genes. This is a type of experiment that's called a screen, because you're just rapidly screening through many different options to see what comes out, basically. And this type of experiment is like totally not feasible in mice. It's not at all feasible in most other organisms, but flies are so cheap and so fast and so easy to work with that you can kind of get away with that. So that's one answer, which is that people who work in Drosophila tend to just test many different genes all at once. The reason why I'm interested in these particular cell-cell contact genes, part of this comes from what we know about the adult ovary. So again, I'm focused on the larval ovary, which is like the developing organism, but we know a little bit about some of the genes that control the survival of egg stem cells in the adult. And so some of the genes that control the survival of egg stem cells, I want to test if they have a role in in producing the right number of egg stem cells during development. So to find this list of genes, I went into the literature and I read a bunch of papers about what we know about egg stem cells, and basically pulled out those genes and decided to test them in my system. So that's a little bit of the more specific answer for me.
And actually, this gets into a really great question that I think was a top question for people, which is like, what happens when you've done research? Like, where does it go? Mm-hmm. And the sort of like, basically the place that research goes, once you have done research that like characterizes a system enough, where you've looked at it from multiple different angles and you have careful controls and you like, according to your field, have sort of like substantially enhanced our understanding in some way, you will then publish it in a scientific paper. So Mikala mentioned she actually went through the literature and she went through papers that were published to sort of like find more information about genes that she could test. And basically, those published papers are the result of people doing very, very careful work. They then submit it to a scientific journal. It then undergoes a process of peer review where experts in a field will come together, they will read a paper, they will say, hey, I think you should do more experiments to better support your hypothesis that you're putting forward, or hey, have you thought about this thing and addressed it? And then ultimately it goes into a journal. Mr. Woodbury mentioned this in first period, but a big issue that I think a lot of scientists have with the process of publishing a paper in a scientific journal is sometimes scientific journals will then hide them behind paywalls for the general public. So they might ask you to pay $40 to access a research paper, which we as scientists hate. And like there's been a lot of discussion in the scientific community about changing that system. But one thing I will say is there are journals that are increasingly moving towards making all papers free for everyone to access. And two, and Mr. Woodbury mentioned this, and thank you for doing so, if you email the authors on a paper that you would like the paper for free, they will almost always send it to you. So if you find a scientific paper that you would like, I would suggest you email. A lot of times there's like 10 authors under a paper because science is very collaborative. But I would suggest you email 2 people. You email the last name on the list and you email the first name on the list. The last name on the list is the professor. So Mikala mentioned she's in a professor's lab. And so there's the professor, and the first author will usually be the person who did the majority of the work. So Mikala will be the first author on the paper where she publishes the information that she's finding. A little bit of, maybe too much detail, but just wanted to go into like, what does the process of scientific publishing look like? Mikala, there has been some cool stuff popping up on the screen. Are you in a place where you can describe some of what we're seeing?
Yeah, so I'm going to talk a little bit about something that I noticed in the image. So I was like going through, and as you can see here, basically this slide bar and this like little number helps me keep track. I told you that we were taking 207 different images. This tool lets me keep track of which image I'm looking at out of the 207. And so as it was going through and updating, I noticed that, so it's gonna load a little bit slowly, but around, here it is. Okay, so you guys can see slice 190 out of 207 looks good. And then slice 191 looks a little funky. And that's because the scope got like bumped briefly.
That's because I bumped the scope.
See about taking responsibility?
Yeah.
It's totally fine, but basically just the scope was like bumped while the image was being acquired. And so even though it was, trust me, in the room, it was the gentlest of taps. But this like machine is so sensitive that it picked up that tap and it messed up when it was acquiring the image. And then if I go to slide 192, everything's fine again. But I am that picky. And the way that I need to analyze these images is that picky. And so that one slice, I wouldn't trust the data that comes from this anymore. And so all I did is just restart the acquisition. So we're back on the same sample. We've just started over.
And our friendship is ruined.
And our friendship is ruined. Yeah, that was the end. So luckily I've done a lot worse to this scope.
Excellent.
And so now we've started back over. We're now imaging slice 170 out of 207 or 200 and something, 200, yeah. And so, yeah, we've just started over. So this is a really great example. I'm really glad that this didn't work perfectly for you guys because I troubleshoot all the time when I am imaging. Taking these images is really like a very like active mental process. And so it's a lot of times like this where in order to get really good images and really good data, you have to be kind of using all of your like senses and observing everything that's going on.
Taste is a really important
Taste is a really
sense.
vital one here, yeah.
Yeah, I
Well,
think you guys were also mentioning like in my first period class, like it took you like 6 weeks to kind of figure out, well, why am I not getting like a really good image and everything? Do you think you can talk more about that so my third period class can hear about it?
Yeah. So in the previous class, we talked a little bit about I was going through this period of time where none of my experiments were working, and so essentially, okay, so I'm going to back up and tell you really quickly that it's really important that the green signal here, and that's marking again the protein traffic jam, which sits in the nucleus. What else is in the nucleus? The DNA. And so it's really important for me to see that the green signal is overlapping with the gray signal, because they should be in the same place. Basically, the computer should be able to go through, image all 207 slices in one color, and then go back to the top, and the sample has to be in the same place so that when it images the next color, the images basically perfectly overlay on top of each other. And so what I'm looking for here, and what I'm very happy, what I see successfully, is that the green is overlapping on top of the gray, which means that the image and the sample are in the exact same place as they were a minute ago. But for about 6 weeks in the fall, none of my experiments were working. Let me see if I can actually pull up an example of what this looks like, because I have all of these images saved. Oh yeah, they have great titles like Offset Bad. And what you can see here, so this is an example image that I took from a really long time ago, but as you can see here, there is a lot of green where there is like no gray. And sorry, this is changing back to blue, but as you can see here, here is a bunch of green and there's no gray underneath. And this was a really bad one. This was like the worst offset I've ever seen. And so what this is basically saying is when the microscope was imaging the gray sample, the sample was in one place. And then by the time it started imaging the green sample, the green channel, the sample had moved. And so that was really bad. And so this was a period in the fall where like none of my experiments were working. And it ended up being that I had to do a lot of troubleshooting to figure out what was going wrong. And it ended up being a combination of 3 different issues that were all kind of contributing and like producing the same problem. Yeah, so I couldn't do any experiments for a couple of weeks, and it was really sad.
Could you share the silliest of the issues?
Yeah, I think the silliest of the issues was the temperature of the room was no longer the correct temperature. Basically, this problem started happening after our building switched. It was like in the fall. So it happened after the building switched from air conditioning to heating. And the temperature of this room accidentally got hard locked by facilities at like 65 degrees. And this microscope, this machine produces a lot of heat when it starts working. And so because the room was all of a sudden too cold, when the microscope started working, the temperature of the room increased too quickly. Heat causes metal and plastic and glass to expand. And so because the temperature wasn't being properly regulated, the sample was like expanding and the glass slide was expanding and everything was moving around. And
when you're looking at 1/100th of a millimeter, metal expanding is a huge problem.
Yeah. So that was one of the problems. Another problem was that the glass slide that sits in the microscope itself was moving. And then I also think that the sample on the slide was also moving a little bit as well. And so it was like the combination of all of these things that was resulting in this type of image, offset very bad. But this image that I'm producing here, this looks fantastic. My membrane looks really nice with this red signal. My green is perfectly on top of the DNA. So I'm very happy with that.
What other questions do people have?
Do you have a question, Corey, or yeah, okay. Yeah, we got another question over here.
So, is like the purpose of using like the green, like tracking the traffic jams, is that to ensure that like the samples that come after the green are still like accurate or like in a line? Like, is it almost like a safety net for the rest of your imaging?
Great question.
That's a really, really, really good question.
And I actually asked Mikala the same question after our first session.
Yeah. Jess was like, oh, so the traffic jam is kind of serving as this like internal, like what could you call it? Like
Which
an
is exactly
internal
what you said.
control,
Yeah.
kind of.
Which is exactly what you just asked.
Yes, so overall, no. I'm actually interested in the traffic jam positive cells as well. But it turns out a sort of helpful coincidence that I have this really easy to tell indicator of when the samples are offset from each other. So I didn't intend for it, but it is very helpful. I will say that actually all of the channels, all of the different colors that I look at can serve as indicators of this shifting. As you can see here with the red signal, it should be like in between the nuclei. Oops, I'm gonna stop the acquisition here. It should be like the red signal should fall in between the nuclei. And when the offset is really bad, those lines are just crossing down the middle of the nucleus. And that also means that the sample has been offset. So they all actually will tell me if there's something wrong, but the traffic jam is like the earliest indicator. Yeah.
Good question. What else have you got?
I think my question is like the sample that you're looking at with all those cells, is it just like a singular level of cells, or do you also have some cells like kind of piled on top of that too on that z-axis?
Yeah, so it's actually like a really complex organ, and there are cells at like every point in this Z-stack. So if I go to the very top of the Z-stack here, this is like the outer sheath of cells that surround the ovary. And then as you start to go deeper and deeper, you start to see, remember these are the like bright dots of the HTS that mark the egg stem cells as well. And so the deeper you move into the ovary below this sheath are my egg stem cells, and then that population continues through a lot of the depth of the tissue, and then at the bottom, there's actually egg stem cells all the way through the side here. So yeah, I actually don't know how many cells there are in each ovary, but there are probably about 1,500 cells in each one. Probably.
And I guess probably also depends the time that you image.
Yeah, it also depends on like the developmental stage of the sample that I look at. If you look at a really early developmental stage from a really, really tiny baby larvae, it probably only has 100 cells, maybe even less. And then later into development you go, the cells have done a lot of dividing and a lot of growing, and the organ is now much bigger.
Yeah. This actually brings up just like one thing that I wanted to highlight. Oh.
So we're done. The image has successfully acquired, and so it just kind of, the program just let me know like it's finished. It combined all of the channels and presents them to me all at once, which is really nice. And this is a really clean, crisp image. I'm very happy with this one. So what I'm gonna do now is I'm gonna do that same process that I started off with. I'm gonna find a new sample. I'm gonna set the Z stack to find the top and the bottom, and then I will be back.
Lights back on?
Oh, it's fine.
Oh, you're good?
Lights off, yeah.
You operate in the dark?
Operate in the dark.
Amazing. So one thing that someone asked a great question in earlier class, which is like, the stem cells here, are they very similar to human stem cells? Like, are you interested in sort of studying how they develop? So you can like understand how human stem cells develop, and then you can take those human stem cells and like use them as replacements, maybe for neurons or things like that. And it was a great question, and I'm gonna provide my answer, and then Mikala might chime in with some of her own thoughts as well. Mikala's science is super fundamental, and so it really is not necessarily like, how can we understand these egg stem cells so we can understand human stem cells? I think, and again, Mikala's gonna correct me if I say something way off base, but it is really about like, how can we understand how stem cells develop in the context of a really complex organ? Something that I forget which class pointed this out yesterday, but we have sort of divided these two cell states into egg stem cells and non-egg stem cells. But actually, the non-egg stem cells are divided into even further groups of cells. Traffic jam is actually only marking the nucleus of some of them, which are called interstitial cells that Mikala's really interested in. But without getting too much into the weeds, I think like really like the core of this project is like, how does a very complex organ like a liver or a heart like fully develop? And there's really no way to do that with human cells right now. And we wouldn't even sort of know where to start, right? Whereas with flies, as Mikala mentioned, there's a lot of things you can do with flies because they so rapidly develop. And there's a lot of core concepts in the work that Mikala's doing here that we can ultimately use to better understand how like human organ systems are developing. I think also there's some links to human fertility as well, because these are egg stem cells. And so thinking about also how do human eggs develop is an important thing.
Yeah.
Anything to add on?
No, I think that's all accurate, and I would definitely stress the importance of the developmental of the model organism to my research. You know, there's some work that you can do with cells that are isolated and in a dish, but you can learn much more about how these complex systems develop by studying them inside the organism. Okay, so we've set our I've set a new Z-stack, I found the ovary, I've made sure that all of the laser power is right, and so now I'm going to start this final experiment. And so this is just the same thing again. We're starting just imaging the DNA at the very top of the ovary. We're gonna go from top to bottom, looking at DNA, and then top to bottom for traffic jam, HTS, and finally our VASA. So I do this all day, pretty much.
I think we have like 10 minutes left. What other questions do people have?
Yeah, we got a question over here.
It kind of makes me think of the lab that we have recently and how they're starting to learn like organic and working on that. So like this stem cell research, could it possibly in the future be translated to something like that? I don't know, this is just really kind of crazy, but like growing organisms per se.
I am so sorry, but we're gonna need to ask for either the question asker or Mr. Woodbury to repeat for us.
Yeah,
You want to come up here a little closer to?
yeah.
It
There's
is a
a really loud fan.
Not your fault, it's our fault.
Oh, it's all good. It's a good question though.
So, I recently heard about the lab-grown meat that they were doing, and I was just wondering how possibly in the future this stem cell research can be translated to like organ development.
Yeah.
Yeah.
We're
it's a
getting
really
into
good
development.
question.
Yeah, good question.
Yeah. So I guess that there are a lot of different possible ways that stem cells can be used to help and advance human health. But I'm gonna give one example in particular. This was a really exciting paper that came out a couple of years ago. And they found that for people who have type 1 diabetes, there is a missing cell population in an organ called the pancreas. And the cells that are in the pancreas are really important because they produce insulin, and that helps you regulate the amount of like glucose in your blood. And so what these researchers were able to do was actually transplant stem cells into a patient and have those new stem cells produce these cells that the pancreas was missing. And then those cells were able to produce insulin and help manage their diabetes. And so this represents a really exciting possibility for impacts of stem cells on human health. This therapy is currently like very expensive and still very, very experimental, but it represented this like incredible advancement in our ability to use basic science to actually help with human health. And this came from a lab that had been working on the fundamental science of type 1 diabetes for like 20 years. And because of all of the basic science that they knew, they were able to develop this therapy. And so there are now a bunch of different attempts to do the same thing for many different systems. Your liver has an amazing ability to regenerate. That's why you can actually donate your liver to someone who needs one. And what they do is they just cut your liver in half and your liver will regrow. But we would like to be able to make this process like more robust or actually even develop like synthetic organs. So there's a lot of the power of organ development comes from stem cells.
Yep.
And so there's a lot of research being done into how these stem cells can be used for human health. That's just one example.
That is a great example, and I want to add on one of my own. I think when I came to your class, I presented the fact that I study collagen, and our lab has actually adopted a collagen organoid where you can grow collagen from stem cells in a dish. And one of like developing these protocols where you can grow organs in a dish, and I will say the collagen organoid is not to replace collagen in people. It is actually used then to screen drugs to treat diseases of collagen. But the thing that I'll say about that is figuring out these organoid systems to make where you're sort of growing organs in a dish requires an understanding of also how the organs develop in the context of the whole organism. And we were just talking about this with Mikala's work, but there's been a sort of classical view of differentiation of stem cells into their final cells, which you add hormones and you add sort of different extracellular factors that encourage differentiation. But for the collagen organoids specifically, one of the findings that they realized in order to make them differentiate into a collagen organ in a dish is that you had to add pressure and fluid pressure. And that came from studying how collagen developed in a model organism, like the one that Mikala's doing. A lot of thoughts that we have surrounding this.
Yeah.
Thank you for the question. Very, very good. They're all good questions.
Yeah.
What other questions do you have?
Anybody else got any other questions? Don't be shy. Looks like we got none so far.
All right, I think there's a couple that came up. How would you recommend people start doing research? Actually, so I think Mikala introduced a little bit of her research discovery. I feel like Jamika's answered this question a couple times in the context of some of the other sessions that we had. Jamika, would you be comfortable sort of talking about your research journey and your big recommendation for people who want to do research? I will say Jamika's also a scientist. I think we introduced her as such previously, but she studies astronomy and she works with major telescopes.
The opposite end of the spectrum, microscope tells me.
Yeah.
Yes, I'm so grateful for the experiences that I've had and for all of the amazing people who have made that happen and who have helped me and mentored me and shared their knowledge with me throughout this journey. Yes, I'm a non-traditional student. I was a teacher for 13 years. And then after that, I went back to school to work on my third degree, which I'm currently doing. This is an undergrad degree in planetary sciences here at Hilo. And I, as an undergrad student, I did not have opportunities for research. And so it wasn't until I started just letting people know that I'm interested and that even though I wasn't an upperclassman who were doing these kinds of getting these research experiences like the year before they graduated or their junior year, I let professors and just other astronomers who I worked with know that I was seeking these opportunities and that I'd be grateful for any support in making that happen. And I'm so grateful that more than one person said they were willing to help me. And I'm grateful to be working with one of those amazing people right now, Dr. J.D. Armstrong of the Institute for Astronomy at University of Hawaii. On Maui, doing asteroid research. I'm at the very beginning stages of it, and I'm learning so much. But it wasn't until I reached out and asked questions, asked someone, and let them know, hey, this is what I want to do, that I finally got those opportunities. Yeah.
Yeah. And I will just a plug, and Jamaica, correct me if I'm wrong, but you actually do Shadow the Scientist sessions that are open to the public, where anyone who wants to see you collect data on a telescope can join in and do exactly what we're doing here, but with you on these really, really cool telescopes, right?
Absolutely. We're so grateful to have collaborations with the Keck Observatory, the International Gemini Observatory, Cairn Francois Telescope, the NASA Infrared Telescope Facility. I know I'm forgetting someone, but the observatories on Mauna Kea. And in fact, I had 4 Keck observing sessions this week. So yes, and then I have a Gemini observing session this Sunday. So these are always available. If you just check out our website, they're typically there for you to register. And we also do have sessions in other fields too, including ecology, oceanography, volcanology, and we'll be starting hopefully soon with some gravitational wave sessions. I'll be visiting LIGO in a few weeks. So we're bringing them online hopefully as well. And as far as astronomy, I forgot that we do also work with the Chara Array, which is right there in California, as well as Lick Observatory. So we're happy to have you in any of those sessions. And Mr. Woodbury, please feel free to email me about that, or your students, pass my email to them. And we do dedicated sessions just like this for students where you all are the audience, and we can tailor that time as well. And we work with Las Cumbres Observatory, which has a network of robotic telescopes around the world. So we can actually do that observing during the day, and you can work with data and do projects with one of the astronomers if you're interested. So yeah, feel free to reach out to me about that if you're interested.
Well, I think
So
we got a little less than a minute left, but I do have one more question from a student, if
yes.
that's okay.
So when you publish your work into journals and stuff like that, do you have to like get a copyright or a patent for it? Like, how do you protect your work as a scientist?
Great question. You, the whole point of doing scientific work usually is that it's free and available to the public, and then anyone should be able to do it. There are people who will get patents or copyrights for very specific techniques, if they want to go on and use them in biotech. I would say engineers really like to get patents.
Yeah.
Biologists, we really like to share our work and then make it available to anyone.
Yeah. So basically, the sort of final product is that you publish your work and then it has your name attached to it in like a scientific journal. And then when other people want to like use that research, they like cite you and give you credit for the work that you did. That's
Yeah.
kind of the model.
Yeah, and just to add on one thing to Jamaica's point about finding research, you at any finding research, one of the best ways to do it is in an institution, like an academic institution. For those of you who are going on to undergrad, if you want to find research, I really want to just emphasize what Jamaica mentioned, which is to ask professors to work with them. And professors often get asked. You will often get a lot of no's. I think I emailed 15 professors before I found a research lab that I was able to work in undergrad. But that is really, if you're interested in research in any discipline, you have to ask.
Yeah, just cozy up to the professor, like go to office hours, talk about their class or their subject if you're taking it, talk about their research, and then once they put a name to a face, yeah, it just increases your chances of getting to work in their lab.
Absolutely. Okay, I'm gonna turn the lights on just so you can see us maybe a little better. But
Yep.
Thank you all so much. This was a lot of fun for us out here in Boston. And yeah, I hope that you enjoyed this session on the scope.
And thanks again, Mr. Woodbury, so much for letting us come into your classroom. Thank you, Jamaica, for putting all this on. Thank you, Mikala,
Yeah.
for letting, sorry for bumping your sample.
No, it's all good. The image is, the experiment's done, so perfect timing.
Yeah. Please, Mr. Woodbury, please email me if you have any more questions from students. Happy to answer.
Wonderful. Thank you guys so much. I feel like this is very special for my students, and they got a lot out of it. So yeah, we're definitely going to be recapping this tomorrow and next week. And I just want to say thank you guys so much for this opportunity. This is wonderful.
Thank you.
Pleasure. Yeah.
Thank you.
Bye.
All right, you guys have a good rest of your day.
You
You
too.
too. Bye bye.